Mycobacterial pathogenic strategies remain poorly understood. The slow growth rate of most species, the impenetrable nature of the cell wall, and the hazards of working with pathogens make mycobacteria difficult to study and are largely responsible for our poor understanding of these organisms. In this video, we will demonstrate the technique of electroporation, which involves subjecting cells to a brief and high electrical impulse which allows the entry of DNA. It is the most widely used method for introducing DNA in mycobacterial cells.
Hi, I'm Renan Goude from the laboratory of Tanya Parish from the Center of Infectious Disease at the Barts and the London School of Medicine and Dentistry. Today we will show you a procedure for electroporating mycobacteria. In our lab we study Mycobacterium tuberculosis, but because it is highly pathogenic and requires a Cl3 containment facilities, we will show you this technique in the non-pathogenic Mycobacterium M. smegmatis. So let's electroporate some bacteria.
To electroporate mycobacteria, we need to make electrocompetent cells. It is important to remember that pathogenic mycobacteria represent an important biohazard. Therefore, all culture and genetic manipulation must be carried out in an appropriate containment facilities inside a Class 1 safety cabinet. Risk assessment must form the first part of any experiment with pathogenic mycobacteria. To make these, we start with the culture.